Okay, so first we have chapter 1, 10, and 11, which is the genetic material, 2, opposing viewpoints, why and how the experiment, they believe protein is the genetic material, how they found out DNA is the genetic material. So in the 1940s, proteins and nucleic acids were major candidates for genetic material. However, geneticists favor the hypothesis of proteins being the genetic material. This was because proteins were diverse and abundant in cells. Additionally, the tetracleotoid, which is spelled TETRANUCLEOTID, hypothesis, which stated that DNA contains equal amounts of 4 nucleotides, also influenced scientists to favor proteins as the genetic material. In other words, they believed that DNA lacked the chemical diversity since there were only 4 nucleotides. Thus, they suggested that it could not store extensive genetic information. However, experiments began showing evidence that DNA was in fact genetic material. The Griffith, which is spelled GRIFFITH experiment, was an experiment which observed the avirulent and virulent strains. And virulent is spelled AVIRULENT, and then virulent is spelled the same but without the A. Strains of Diplococcus pneumoniae, which is spelled DIPLOCOCCUS, and pneumonia is spelled PNEUMONIEA. When mice were injected with the avirulent strain, they remained healthy and lived. However, whenever mice were injected with the virulent strain, they died. Griffith then he killed the virulent strain and injected it to the mice, and the mice lived. Thus, he hypothesized that if he mixed living avirulent strain with a heat-killed virulent strain, the mouse would live. However, when the experiment was performed, the mice injected with the mix died. Griffith analyzed the tissue of these mice and recovered living virulent strains of Diplococcus pneumoniae. His experiment showed that virulent strains of Diplococcus pneumoniae could be transformed into virulent. This experiment provided the foundation for the Avery, MacLeod, which is spelled MACLEOD, and McCarthy experiment. The Avery and MacLeod and McCarthy experiment provided the first direct experimental proof that DNA is the genetic material. And the experiment, filtrate, which is spelled FILTRATE, recovered from the heat-killed virulent strains, was treated with protease, which is spelled PROTEASE. It was also treated with ribonuclease, which is spelled RIBONUCLEASE, and it was also treated with deoxyribonuclease, which is spelled DEOXYRIBONUCLEASE, which are enzymes that degrade proteins, RNA, and DNA, respectively. The treated filtrates were then used to transform virulent strains. The protease and ribonuclease treated filtrate. The filtrate that was treated with protease, which is spelled pro and then T and then like tea you drink, and then SE and then ribonuclease, showed transformation, while the deoxyribonuclease ribonuclease-treated filtrates show no transformation. This proved that the transforming principle was DNA and not proteins. Thus, DNA was the carrier of genetic information needed to transform the different strains. Next, Hershey and Chase performed an experiment in which they used radioisotopes, which is spelled radio like Lana Del Rey's radio song, isotopes, radioisotopes of phosphorus -32. Phosphorus is spelled PHOSPHORUS, and also sulfur, which is spelled SULFUR-35, to label the DNA and protein coat, respectively, of a bacteriophage, which is spelled bacteria, but instead of an A, it's an O, and then PHAGE. The bacteriophages were then allowed to infect unlabeled bacteria. The bacteria were observed during the infection process. It was found that the labeled DNA was introduced into the cell while the labeled protein coat remained outside. Additionally, the progen, which is spelled PROGENY, of the P32 labeled phages, which is spelled PHAGES, were also labeled with P32, while the progen, which is spelled PROGENY, of the S35 was not labeled. This provided conclusively that viral DNA alone contains all necessary genetic information for the production of mature viruses. Direct evidence also supported the hypothesis of DNA being the source of genetic material. For instance, there is a close correlation between haploids, gametes, diploids in regard to the amount of DNA and number of chromosomes. However, no such correlation between gametes and diploids exists for proteins. Chromosomes is spelled CHROMOSOMES, and diploids is spelled DIPLOIDS. Gametes is spelled like game and then TES. Additionally, the molecule serving as genetic material was expected to absorb a mutagenic, which is spelled MUTAGENIC, UV wavelength at 260 nm. DNA absorbed UV at 260 nm, while proteins absorbed UV at 280 nm wavelength, a wavelength at which no mutagenic effects were observed. This also answers my Chapter 10 evidence that genetic material is DNA and experiment in bacterial and phages transformation. So that's all together. And then we have Chapter 2, chromatins, nuclease, nucleose, checkpoint, homologous chromosomes, sister chromatid. So chromatin is spelled CHROMATIN, and it is uncoiled DNA. The nucleus, it is found in eukaryotic cells. An abbreviation for eukaryote would be EUK, and it houses genetic material. The nucleoid, which is spelled in UCLEOID, is found in prokaryotic cells, and an abbreviation would be PRO. And it's not membrane bound. And then nitochore, which is spelled KNIETOCHOR, they are an assembly of multi-layered plates of proteins associated with the centromere. The centromere is spelled CENTROMERE, where the spindle fibers bind to the centromere. The cycle checkpoints. G1/S checkpoint monitors the size of cell and the condition of DNA. The G2/M checkpoint monitors the cell replication is complete and monitors damaged DNA. The M checkpoint monitors spelled ITORS. So it monitors formation of spindles fiber system and attachment to the kinetochores, which is spelled KINETOCHORES. And then we have homologous chromosomes, which is like homo, and then log, you log onto something, and then us. Chromosomes, these are chromosomes that are similar but not identical. They may carry genes. They may carry genes for the same inherited characteristics but different alleles. And then we have sister chromatids. And sister chromatids are identical copies of a chromosome attached by a centromere. Centromere is like CENTROMERE. Let me check. And then we have phenotype, genotype, alleles, and genes. Alleles are variation of a given gene. Gene is a unit of inheritance. Genotype is the set of alleles for a given trait. Phenotype is the expression of the genotype, the observable trait. And now we have homogeneous and heterogeneous. Homogeneous and homozygote, both alleles are the same. Heterozygous and heterozygote, the alleles are different. We have now that was chapter 3 and 2. Okay, now we're on chapter 3, correlation between the mechanics of meiosis and the medial principles. So for this we have a little diagram. A says unit factors and pairs, and in parentheses it has the first meiotic protophase, which is spelled, meiotic is spelled MEIOTIC, and then prophase is spelled PROPHASE. And then it says homologous chromosomes and pairs, genes are part of chromosomes. And then for B, it has segregation of unit factor during gamete formation. Gamete is spelled GAMETE. And then in parentheses it has first meiotic anaphase. And then it also says homologs segregate during meiosis. And then homegirl wrote, sister chromatids stay together. And then underneath the first meiotic anaphase, it says not anaphase 2, because in anaphase 2, identical sister chromatids were as an anaphase. Anaphase 2, they have identical sister chromatids, and in anaphase 1, they have homologous chromosomes, which means similar alleles. And then for C, it says independent assortment of segregation unit factors. It says in parentheses following many meiotic events. And then it also says non-homologous chromosomes assort independently. All possible gamete combinations are formed with equal probability. And now we have chapter 4, recessive, dominant, and complete. Recessive or dominant. One allele is dominant over the other. Example, Mendel's pea plants. Mendel's is spelled Men and then DEL. In complete dominance, neither allele is dominant, resulting in an intermediate phenotype. An example is the red snapdragon crossed with the white snapdragon. It results in pink F1 generations, and F2 generation is a 1:1 ratio. Codominance. Neither allele is dominant, resulting in both alleles in a resulting in both alleles in a heterozygote being clearly evident. Multiple alleles of one gene may exist. An example of this is ABO blood type. A/B antigens present on the surface of red blood cells. 3 alleles of a single gene. A/B antigen are codominant to each other, and A/B are dominant to O. Now we also have recessive and dominant alleles. Recessive lethal allele, a homozygous recessive individual will not survive due to a mutation of a gene product that is non-functional. In parentheses, it says no essential product made for survival. Note that the time of death will depend on when the product is essential. Example, early childhood or until adults. Dominant lethal allele is the presence of just one copy of a allele results in the death of an individual. An example of this is Huntington's disease due to a dominant autosomal allele of capital H. This allele is rarely observed because affected individuals must reproduce before the lethal allele is expressed. If affected individuals die before reproducing, the mutant gene will not be passed to future generations. And then we have chapter 4, epistase with examples. Epistase is EPISTASIS. Epistase is an expression of one gene masks/modifies the effect of another gene pair. First, we have recessive epitasis. When a recessive allele at one genetic loci masks the expression of the alleles of a second locus. Recessive allele lowercase a will lead to recessive phenotype regarding the second locale. And locus is spelled LOCUS, and loci is spelled LOCI. And then 3 points are lowercase a, capital B, and whichever one, like whether it's capital B or lowercase b. This is the recessive phenotype. And then it says lowercase a, lowercase b equals recessive phenotype, and the ratio is 9:3:4. Dominant epitasis. When a dominant allele at one genetic loci, which is spelled LOCI, masks the expression of the alleles of a second locus, which is spelled LOCUS. Dominant allele A will lead to dominant phenotype regardless of second loci, which is spelled LOCI. Capital A, and whichever other one, doesn't matter, capital A or lowercase a, and then capital B, and doesn't matter the second allele, is a dominant phenotype. Capital A and either capital or lowercase a, and lowercase b equals a dominant phenotype. The ratio is 12:3:1. Double recessive. Any of the two recessive alleles suppress the effect of dominant genes. Recessive allele lowercase a, or lowercase b will result in recessive phenotype. Example is capital A and either capital A or lowercase a, and lowercase b equals recessive phenotype. Another example is lowercase a, capital B, either capital B or lowercase b equals recessive phenotype. And then we also have lowercase a, lowercase b equals recessive phenotype, and the ratio is 9:7. And we also have double dominant. Any two dominant alleles suppress the effect of recessive genes. Dominant allele A or B results in dominant phenotype. An example is capital A, and then dash, because it doesn't matter which other one, capital B, and then dash, doesn't matter which one, dominant phenotype. Another example is capital A, dash, lowercase b equals the dominant phenotype. Another one is lowercase a, capital B dash equals dominant phenotype, and the ratio is 15:1. And it says novel phenotypes, novel spelled like Novel. The ratio for that one is 9:6:1. Still on chapter 4, we have sex-linked. Sex-linked genes present on sex chromosomes that exhibit patterns of inheritance. For example, color blindness. X chromosome linked, therefore mother will pass it to all sons but not all daughters. Lethal X-linked recessive disorders, absorbed only in males because females can be either heterozygous carriers who do not develop the disorder. Example, Duchenne, which is spelled Duchenne, muscular dystrophy, which is spelled Dystrophy. It says onset prior to age 6, lethal around 20. We have sex limited. Sex limited is expression of specific phenotype is absolutely limited to one sex. Example is feather plumage and chicken. Plumage is spelled Plumage. And then also cock feathering controlled by recessive allele, only expressed in males. It is found on autosomal chromosomes. Auto and then SOMAL. We have sex influenced. Sex influence is one sex is more likely to get it than another. An example is baldness, mostly seen in males, found on autosomal chromosomes. We also have penetrance and expressivity. Penetrance is spelled Penetarnace, and this is the percentage of expression of a mutant phenotype. And then for expressivity, it says one genotype can express quite a few phenotypes. Example, eyeless mutation, the eyeless mutation in fruit flies. Phenotype ranges from either the presence of normal eyes to the absence in one or both eyes. And then we also have anticipation and imprinting. Genetic anticipation is a genetic disease has earlier onset and increased severity with each succeeding, and succeeding is spelled SUCCEEDING, generation. An example of this is meiotic dystrophy, which is spelled MYOTONIC, and the dystrophy is spelled DYSTROPHY, and the abbreviation for this is DM1. And then genomic imprinting is selective gene silencing that impacts phenotypic expression. An example is silencing depends on parental origins of genes, and it occurs in early development.